Hey folks, welcome back. Today we're going to talk a little bit about what can go wrong if the cell cycle does not go the way it's planned. We're going to look today at what is called cancer. You all know about cancer, but how does that work? How does that develop over time? We're also going to look at cell cycle regulation. How is the cell cycle regulated? How do cells know when to perform mitosis and when to not perform mitosis? So the regulation of the cell cycle, very important. And we'll also look at a concept known as differentiation. That is, how does one cell, how does an organism that starts as a single-celled zygote eventually become an organism that has hundreds of thousands of specific cells like bone cells or muscle cells or skin cells? So we're going to look and examine those topics today in this video lecture. So, our objectives today, you should be able to explain how cancer relates to the cell cycle. How does cancer form, and how does that specifically relate to things like mitosis in the cell cycle? You should also be able to understand how the cell cycle is regulated and also to determine the importance of stem cells. We've heard a lot of things about stem cells in the past, what they are, what they can do, and some of the controversies associated with them. So our goal is to go through and talk about these particular topics and hopefully give you a better understanding about what each of these are. So, normally the cell cycle is a cycle, essentially. And there are several checkpoints along the way to make sure the cell cycle occurs without mistakes. It's very much like writing a paper in English class. You have an outline, and you want to make sure to check to make sure the outline is correct. And then you have a rough draft, and you go through and you check that to make sure that everything is written correctly, and if not, you make corrections. And then you turn in your final copy. The cell cycle is very similar, where there are certain checkpoints to make sure that the cell cycle occurs without making mistakes. So there's a checkpoint for spindle assembly. There's a checkpoint for cell size and growth factors. There's a check to make sure that the DNA is replicated properly. So there's several different checkpoints to make sure that this happens. Proteins called cyclins bind to enzymes called cyclin-dependent kinases, or CDKs, to start various parts of the cell cycle. Now, there is much more detail associated with this, but you just need to understand that cyclin and CDK determine whether or not the cell can pass on to the next part of the cell cycle. They're kind of like the checkpoints, if you will, to make sure that the cell has done what it needs to do in order to move on to the next phase of the cell cycle, whether that be G1, S, G2, or the phase of mitosis. Cyclin and CDK initiate several parts of the cell cycle. Again, G1 of interphase is an important one to initiate. S phase to make sure that the DNA has replicated properly. If everything's good, we can start performing mitosis, and if mitosis occurs correctly, we can go through cytokinesis. So cyclin CDK concentrations really help determine the quality. It's a quality control mechanism for the cell to make sure that the cell is going to replicate properly and produce two genetically identical cells. Remember, we're talking specifically about mitosis here, so we need to make sure that all of those cells are genetically identical to each other. And cell cycle regulation through cyclin and CDK helps us go through that and understand that better. When the cell cycle goes right, everything is great. But when the cell cycle goes wrong, we need to take a look and get a better understanding of why this is going wrong. And when the cells grow uncontrollably, where there is no cyclin or CDK to have those particular checkpoints, we call that cancer. And typically when there's a lack of cyclin or CDK, there are no checkpoints, so the cell cycle just occurs rapidly without actually going through the checkpoints. It's very much like just writing a paper and just turning it in. A lot of times you're not going to get a good grade on that because you haven't taken the time to go through and check to make sure that everything is done properly. And it's the same thing when making a cell. This uncontrollable cell growth is called cancer, and this is just a failure in the regulation of the cell cycle for a multitude of reasons. There's regulation in the cell cycle in order to correct any mistakes that may be made. But if there is no regulation, those mistakes get through and cause serious damage to the body. This can be caused by many factors, including carcinogens. Carcinogens include anything that causes cancer, such as ultraviolet radiation, including the sun and X-rays, smoking, right, nuclear radiation. There's a long list of things that can cause cancer inside of the body. Most of them you're probably already aware of. So you need to be careful around those certain types of carcinogens and understand that those can cause cancer inside the body. Carcinogens specifically alter the DNA, and this alteration can be in a location that regulates the cell cycle, and this is where cancer starts to develop, is that when there is cell damage without any repair, most of the time, if you look, most of the time the cell essentially commits suicide. It commits what's called apoptosis. That's just programmed cell death. If there's DNA inside of the cell that doesn't work properly, it will destroy itself in order to prevent damage to the body. However, when this happens with cancer, a lot of times that cell damage isn't detected. And so as a result, that cell multiplies and divides. And as a result, we end up getting a large amount of cancer. We get uncontrolled growth inside of the body. As a result, that causes cancer. Cancer can go undetected until millions of abnormal cells are produced. Cancer is very difficult to detect until it's almost too late. So it's very important to make sure to try to avoid as many carcinogens as possible to prevent cancer from happening. There are several types of tumors, and tumors are simply just tumors are simply groups of cancer cells. Malignant tumors have the ability to spread from one place to another. So if there is a tumor that is malignant inside of the body, those cancer cells can break off from the tumor, travel through the bloodstream or the lymphatic system, and end up in other locations in the body. This is by far the most dangerous type of cancer, is one that is malignant, because it can spread to other parts of the body and cause significant harm. Benign tumors don't have the ability to spread. These are ones that typically are cured with surgery. They are removed from the body, and as a result, they don't cause any more harm because they have not spread to other locations. When cancer has spread to other parts of the body, we say that it's metastasized. And that is just the term that is used to describe that cancer cells have spread to other areas of the body. So you probably heard of these terms before, but may not have a clear understanding of what they mean. So now you understand the different types of tumors and the fact that cancer can be very dangerous because it can go undetected until millions of abnormal cells are produced. Cells simply just cannot live forever. And that's a good thing because old cells typically don't function as well as newer cells. So we have a program, the cells have a program called apoptosis. And this is simply just programmed cell death. This is where cells undergo a process where they destroy themselves in order to prevent the older cells from causing problems. This is used for cells that typically don't work properly and typically prevents dangerous cancer cells from reproducing. However, it is when carcinogens are involved that alter the DNA that have a significant impact on apoptosis. And if apoptosis does not work properly, we end up with cancer, which is very dangerous to the body. One important concept to understand is the concept of differentiation. Multicellular organisms such as humans have many different types of cells, including skin cells, red blood cells, bone cells, muscle cells, etc. And these are formed through a process called differentiation. Different cells inside of the body have different activated genes, and those genes determine how the cell functions. Remember that every cell in the human body has the exact same DNA. But the difference between a skin cell and a muscle cell is the genes that are activated within the DNA. And so that activation causes a muscle cell to be different from a blood cell. It has a different function, all right? And this allows those cells to have different functions in the body. So that's how we can start off as one cell and over time replicate to become many cells. But each of those cells has different genes that become activated that result in different types of cells being formed from those cells. It's a really, really interesting process. And this is how stem cells come into play. Because stem cells are unspecialized cells that can develop into specialized cells. Many times, the stem cells can be placed next to cells that are already differentiated. So for example, if we were to take stem cells and place them near a muscle cell, that those stem cells would develop into muscle cells. Hormones that are secreted by the muscle cells end up causing the stem cells to differentiate and become muscle cells. It's a really, really fascinating process. And this can allow us to develop into many different types of cells. They can be obtained from embryos. You've probably heard of embryonic stem cells, the very controversial way. But they can also be obtained from adults as well. Adults have stem cells in their body. This gives hope for patients with nerve damage. Because after you are born, your nerve cells don't replicate quite the same as a lot of the other cells in your body. Once you've caused nerve damage, the nerve cells do not grow back, and that's because once they've reached maturity, once nerve cells are done growing, they arrest in what's called G0 phase. And this is kind of like the purgatory of mitosis. They really don't do much of anything after that. So that's why when you have nerve damage, like when you have a spinal cord injury, you never recover from it because those nerve cells cannot grow back. But if we insert stem cells in there, there's hope for people with nerve damage because those stem cells could potentially grow into nerve cells. And those nerve cells could reconnect the tissue and allow that patient to move again. So stem cells, albeit controversial, could completely revolutionize how we perform medicine. In addition, stem cells can also recreate different organs in the body. So for example, if somebody needs a kidney, stem cells can actually make a kidney. We can actually do that in a science lab, is to make organs out of stem cells. So there's plenty of different revolutionary new concepts available by utilizing stem cell research. So hopefully you understand a little bit more about how cancer relates to the cell cycle and how the cell cycle is regulated, and determine the importance of stem cells. Again, a lot of very important topics that need to be discussed here, and very deep topics like cancer and stem cells. But hopefully with a little bit better understanding, you can make your own judgment as to how these can be treated and used in the future. We'll talk to you guys a little bit later. Make sure to take your video quiz at the end of this lecture. Have a good day, guys. We'll talk to you later.